Hi, everybody. Welcome back to this new workshop series about root phenotyping. I am Anarita. I'm one of the co-founder of Phenom First, but for this season, we have new members. Do you guys want to introduce yourself?
Hello, everyone. Good morning and good afternoon for some of you. I am Maria de Oliveira. I'm studying my PhD at North Dakota State University and working with high throughput phenotyping. Hope you guys have a good day with us today.
Hello, I'm a postdoctoral at John Innes in UK, and I work with roots. I work with the roots of wheat, trying to find traits that can improve soil health. Welcome to the workshop today. I hope you enjoy it, and it's a great learning opportunity.
So I think many of you probably they know about Phenom First, but in case you don't, we will give just a brief introduction about who we are and what we do. So we are Phenom First. We are a learning community that me and Felipe co-founded in 2020, right on top of the pandemic, with the main purpose of bringing together expert and beginners in high throughput phenotyping. So this is a platform that we try to provide free trainings, especially to empower early career researchers, but also all our workshops, they are a live library about resources in phenomics. So if you want to be updated about what we do, please subscribe to our YouTube channel where you are looking at us now, and you will see that there are so many resources. Also, the workshop today will be there, so you can watch and rewatch the workshop whenever you want. So also, if you haven't done it yet and you want to know more about us, so we have our website where we list all the material and what we do. We are on Twitter, and so where we, of course, advertise our events, YouTube, and of course, if you want, we also have a Slack channel. So if you want to join the Slack workplace, it's a very good place where to post questions, and there are many experts in the field. So if you want to join our Slack workplace, you can just send an email to phenomforce@gmail.com. And with this, I think I leave Maya to introduce the speaker of today.
Our speaker today is Benjamin Delory from the University of Leuphana, and he's going to be introducing us to the use of mini-rhizotrons for root phenotyping in the field, from setting the mini-rhizotrons to the pipeline of analysis of all the images. So thank you, Benjamin, for joining us today, and the screen is yours.
Thank you, Benjamin. So, I want just to say to everybody that if you want to ask a question to Benjamin, feel free to write that in the chat. And whenever Benjamin is available, we will ask the question. So with this, Benjamin, good luck, and we will be here to help you whenever you need.
Okay, thank you. So I guess I'm going to start. So my presentation today and the workshop today will be about how we can analyze root distribution using two different types of setup. And I divided actually the presentation in two different parts. In the first part, I will just show you how we use Rhizoboxes in our research group to analyze root distribution in the soil in a very controlled experiment. And in the second part of this talk, of this workshop, I will focus much more on field techniques, on Mini-rhizotrons, and how we can use it to look at root distribution at the community level, but in the field. And for each setup, I will describe you the experimental setup, how we install, for example, the Mini-rhizotrons in the field, how we collect images that we're going to use for root phenotyping, how we extract information from these images using different image analysis pipelines, and finally, what kind of data we can get from it and what kind of graph or figures we can make to answer or research questions. So just to guide us through this workshop, I would like us to think about this research question that we are trying to answer in our research group. And this is to answer this question that we set up multiple experiments, Rhizobox experiment, but also field experiments, and all the information, all the information that I will show you today is linked to this particular question. So I think it's worth mentioning it. So we are very interested in our research group on priority effects, and we are very interested in how the order of arrival of different plant species during assembly can affect the vertical distribution of roots in plant communities. So in other words, if you have some plants, some groups of plants arriving before others, how does that affect root distribution? And to answer that question, we developed and we designed a number of experiments. And so I will start with the first one, which is how we use Rhizoboxes to analyze root distribution of grassland communities, but in quite controlled experiment because it was a greenhouse experiment in that case. And so just so you know, I will just focus on the devices and experimental setup that we use in our research group. When it comes to Rhizoboxes and Mini-rhizotrons, there are actually quite a number of more or less sophisticated setup that you can use. But I would just focus here on the ones that we use in our research group. So first question, what are Rhizoboxes, or also called root boxes? It's actually very simple. It's just a container that is equipped with a transparent front window, and it is filled with soil in the context of an experiment. And we often use this type of device to non-destructively monitor root growth or root system development over time. But like every system that we use in controlled experiment, these Rhizoboxes have a couple of pros and cons. So that means advantages and also kind of disadvantages. So if you look at the pros, Rhizoboxes are quite customizable in design. That means we can actually play around with quite a lot of settings, like the height or the width or the depth of the Rhizoboxes. We can also change the construction material. They are quite easy to replicate because, as you can see, they're not that big on the images on the screen. This particular Rhizobox is only 60 centimeters in height and 30 centimeters in width and 2 centimeters in depth. So it's quite, it's reasonably small, and it's quite easy to do an experiment with around 50 Rhizoboxes at a time. And the advantage of this type of Rhizobox, let's say, is that it's compatible with different types of imaging techniques that allow you to study rhizosphere processes. And I'm mainly thinking here, for example, of planar optodes that would allow you to image pH variations at a very fine scale, at the root scale, pH variation at the rhizosphere level, also oxygen and carbon dioxide concentration. You can also monitor enzyme activities around the roots, which is a technique called zymography. So this type of system has quite a number of advantages, but of course it also has strong cons, and the main one being that you restrict quite a lot root growth. I mean, the plants have very tiny space to grow, and you force the root to grow in a kind of two-dimensional plane. So of course this is kind of a simplistic setup, and it's also quite difficult to track roots of different species, especially if you work with species-rich plant communities. In this picture, we only have 9 different species per Rhizobox, so it's still quite easy at the beginning to trace the root back to the right species, but over time it becomes more and more difficult. So when we use Rhizoboxes in the greenhouse or inside a climate chamber, this is how it looks like. So we have basically plastic containers in which we put between 5 and 6 Rhizoboxes that were filled with soil. And here you see you already have plants growing. And those Rhizoboxes are tilted at an angle, usually at a 45-degree angle in this case, to just try to force the roots to grow along the transparent front window. So the front window is always facing downwards, as you can see here. And we added 2 additional components. At the back here, we added a white polystyrene plate to just avoid, because the Rhizoboxes in this case are black, so they can overheat quite quickly, especially in the greenhouse. So to try to avoid overheating, we just cover them with a polystyrene plate. And at the front side here, for the first Rhizobox, we added a black plastic plate to prevent root exposure to light and try to keep them in the dark as much as possible. And we have this type of boxes that are replicated, I don't know, 10, 15 times in our greenhouse. So this is how the experimental setup looks like. It's quite simple in this case. But what is quite important when we do this type of experiment is to have an image acquisition system that maximizes image quality, the quality of the image, and it will affect what type of information you can extract from them, and also maximize the throughput. That means how many images, how many Rhizoboxes can you image per hour, for example. And so for that purpose, we our technician built this what we call a photo box, where we basically put the Rhizobox that we want to image here in this metallic frame. And inside the photo box, we have a camera holder that holds a camera right in front of the Rhizobox at a distance of around 50 centimeters. And inside this photo box, we have lots of dark fabric to keep everything dark and avoid reflection. And we have two LED tubes that illuminate the front window of the Rhizobox laterally to avoid having too much reflection and having this mirror effect when you take an image. And here in the front, you just put the Rhizobox that you want to image. And we also have a computer that is connected to the camera, and using the live shoot option of the software, we can then take images remotely and see if we have to adjust a few things before taking the image or not. So this is our image acquisition setup, and you have here information about the camera that we use. And if you want really a full description of the image acquisition setup, I strongly recommend you to have a look at this paper that we published beginning of the year. You have the link on the slide. And also provided, actually, this is something that I will put on the chat for the Phenom Force members. Yes, you can download actually the slide of the presentation at this link. So if you could provide it for the participants, Maya, that would be fantastic. Good. So this is our image acquisition setup.
Benjamin, I was also sharing some for the acquisition of the Rhizoboxes themselves. Somebody was asking about the estimated price. Do you have an idea? I have shared the Vienna Scientific, which is a source.
The price of a Rhizobox?
Yeah.
I guess it varies a lot because you can build these Rhizoboxes yourself because it's not that complicated to build, and you have some blueprints that you can find on the internet. For example, the front window can be simply an acrylic, so PMMA plastic front window that you can find easily in a bar market or something like that. So I would say I don't have exactly the price in mind. Maybe this one that you have here in the picture was around 200 euro, something like that. Maybe even a little bit cheaper because we made it. They were custom made by a company in this case. But I think you can have a whole range of prices depending also on the size of the Rhizoboxes you want to use, depending on the material that you're using to build your Rhizoboxes and so on. So you have quite diversity of prices, I think, but you can do it quite in a quite cheap way, I think, as well.
And a question that probably will come up quite soon. There is quite two schools. There is some people that prefer to use perspex because it saves a bit of money. Would you recommend glass or perspex?
In our case, we mainly work with plastic, and because especially with acrylic, so this plexiglass, simply because it has a quite high light transmission. Around more than 90% of the visible light will be transmitted through plastic, for this type of plastic. The only drawback with PMMA or with acrylic is that it can be very easily scratched. And this is actually the material that we use for the Mini-rhizotron tubes. I think that the front windows of the Rhizoboxes that I'm just showing you right now, they are not PMMA. It's another type of plastic. I don't have the name in mind. But as long as it's transparent and you can see the roots, nice roots growing behind the front window, I think it should do the trick. I haven't used personally glass front windows. I think maybe it's a bit more complicated because it's a bit more fragile maybe, and if you want to open the Rhizoboxes frequently during the experiment, it can be a problem. But yeah, I've mainly worked with plastic from Windows.
Excellent. Thank you,
cheaper.
Benjamin. Thank you.
You're welcome. Yeah, so this is the image acquisition. And so with this type of photo box, you get this type of image. So this is an image that is around 60 centimeters in height and around 30 centimeters in width, in which you can see the root system of the 9 species growing in the experiment. And this is a picture that was taken at the end of the experiment, because you see that most of the roots probably start to reach the bottom of the Rhizoboxes, which is usually the point at which we decide to stop the experiment. And the image analysis pipeline consists of 2 main steps. The first step is to try to identify, when you have an image like this, what are the root pixels and what are the soil pixels. This is called image segmentation. So you try to get a segmented image like this, where the root pixels appear black and the background or soil pixels appear white. So you try to simplify the information in your image to isolate the root information that is of interest to you. And to do this, because we have quite complicated images, where when you have roots embedded in a soil background, it's very difficult to use classic thresholding technique to distinguish the roots from the background. And so to do this, we used the software that was developed in Copenhagen by Abraham Smith, so-called Root Painter. And you had there was a phenome workshop last week about it. I will provide the link to the video in a couple of slides. But we use the Root Painter software to train a neural network that is able to segment the roots in this type of image. And when you pass this image through the network, through the neural network, you get an output like this that shows you where the model predicts the roots to be. And here in this situation, you see that the model does actually quite a good job at predicting where the roots in my image are. So this is the first step, segmenting the image. And then the second step will be to try to get information out of this segmented image. By information in this context, I mean root distribution indices, for example, that will tell you how the roots are distributed vertically in your image. And you can extract a number of parameters. I will show you, I think, a couple, the most common ones, and how you can calculate them in the next slide. So, as I said, the first step is to segment the image taken during your experiment to only focus on the roots. And to do this, we use Root Painter. And here you have the link to the Phenome Force workshop of Abraham Smith that provide a really detailed step-by-step explanation on how you can train a neural network, a model, a Root Painter model, to do this task. And I strongly recommend you to have a look at this tutorial, because it's really self-explanatory. Just something that I wanted to mention, because I always think that it's something we have to keep in mind when we do a Rhizobox experiment, is that the root length that we see along the front window of a Rhizobox is not the same as the total root length present in the Rhizobox. So that means that the visible root length is the root length that you estimate based on image analysis, and the total root length on the horizontal axis here is that when you extract all the roots from the Rhizobox and that you measure the length of the totality of roots present in your Rhizobox, you see that you have quite often a quite good correlation between these two variables, but that the slope of this relationship seems to be very species and context dependent. That means for some species, you have quite good agreement between the two. That means you can see quite a lot of the roots, but for some of the species, you see only a small fraction of the roots present in your Rhizoboxes. So it's something that we should always consider. And this is actually a study that was conducted by Kerstin Nagel from the Jülich Forschungszentrum a couple of years ago, but I always find this graph very, very useful. And I think it's important to have this in mind when we do this type of experiment. Okay, so what type of feature can we extract from these segmented images? Once you have them, after using Root Painter, you have these nice segmented images, and now we want to extract information from them. So there are different ways to do this. I will just show you one analysis pipeline that I developed for the context of a research project here at our university. So here are a list of very common distribution indices that can be extracted from this type of image. The first one is called the mean routing depth or average routing depth. And simply put, it's the depth value above which 50% of the root pixels are located. That means above this line, you have 50% of the root pixels in your image, and below this line, you have the other 50% of the root pixels in your image. And what you're interested in is to find out, okay, at which depth this line is located in your image. A second parameter of interest is the maximum routing depth, which is the depth of the deepest row, pixel row in which a root pixel is found. And in here, you can see that because we have some plants that grow quite fast, the maximum routing depth is actually very close to the bottom of the Rhizobox in this case. You also have this called dx in this slide, but this distribution indices that is often seen in root phenotyping papers, like for example D90, D95, etc., which is the depth value above which x%, a certain percent of root pixel are located. So this is similar to the mean routing depth, except that you vary this percentage to another value. The deep root fraction is also an interesting parameter that can tell you, provide information about the routing pattern in your Rhizobox. And this is basically the proportion of root pixels that are located in the lower x% of the filtered image. For example, I usually take x% to be 30%. So for example, what is the proportion of root pixels that are located in the lower 30% of the image? So this is a way to assess the deep root fraction. And you can also calculate in a more classic way, the root surface area, which is the total area of root pixels present in your image. And you can also calculate the relative root surface area, which is the ratio between the root surface area and the size of your image. So it's the proportion of root pixels in the segmented or filtered image in this sense. And it's comprised between 0 and 1. So all these parameters provide information about the root distribution. But now I would like to show you how you can calculate it. And to do this, we actually developed in the context of a research project at our university an R package to do this. It's called POEM-DEMO. POEM is the name of the research project. It's an abbreviation for priority effect mechanism, and demo because it's a package that I developed specifically for this workshop, because it contains only the functions that are relevant for this workshop. Develop other functions that are not necessarily relevant to us in the context of this phenomenon force workshop. And this package is available on GitHub. So you have the link on the slides of the presentation that I just provided. So you can download and install this package. It's open access, no problem. The only thing you need to do, and this is something that, that is important, because the package contains both, was written in both R and Python languages. There are a few lines of code written in Python. So that means if you want to use this package, you will first need to set up an environment to be able to use Python within RStudio. And to do this, I would strongly recommend that you have a look at this tutorial. That is also, if you go on my GitHub page, you will find a link to this tutorial that will provide you really detailed and step-by-step instructions on how you can set up an environment to use Python within RStudio. And when this is done, you can then simply install the package from GitHub. So you just use the install GitHub function within DevTools, and you specify the location of the GitHub repository. And finally, you can use the functions that were developed that are located in this package to analyze root distribution. And one of the tools that is provided within this package, so if you download the package, you will have this tool available to you, no problem. There is a Shiny app that I developed to kind of automate feature extraction from segmented Rhizobox or Mini-rhizotron images. And as I mentioned, it uses both R and Python, so it's quite important that you set up first your Python environment in RStudio. And you can use this Rhizotron Data Explorer to compute all the row distribution indices that I just mentioned earlier in the presentation. And I will just show you how it works. So we're going to try it now. And to do this, you have I provided also a number of Rhizobox images on Zenodo. So you can click on this link in the slides of the presentation, and you will be directed to my Zenodo repository. And you can download here the segmented Rhizobox images. So that means the images that were provided after analysis with Root Painter. And we'll use these segmented images to extract some features that are related to root distribution. And I will show you now how we can do this. Okay, good. So here I have my RStudio session open. The first thing I'm going to do, I will load the Poem demo library. So you can do this, of course, after.
Benjamin, sorry. Can you please increase a little bit the font size in R so they can read?
The font size, yes, I think so. Does this work better like this?
Yes. Yeah, I think yes. Thank you.
Okay, thank you. So the first step is to load the Poem demo package. And I can do this because I've already installed the package and I've already set up my Python environment within R. So it's easy for me at this stage. So I'm loading the package. It's also loading the different dependencies. So we're going to use Shiny, Tidyverse, and other associated packages like Reticulate that allows you to use Python within R and stuff like that. And if you want to use the Shiny app that I just showed you to extract root distribution features, parameters from your segmented images, you just have to run this Rhizotron Explorer command. It will open the Shiny app. It's a bit slow, but that's fine. Just downside a bit. Voila, perfect. So you see, it's a very simple Shiny app that we developed mainly to streamline this feature extraction process when we have Rhizobox images to analyze for our experiments. The Shiny app also has functions to copy, Rhizotron or Mini-rhizotron files, new images from one repository to the other. This is something that we use when we come back from the field, for example. But right now we are interested in this image processing section. So the first thing that we need to provide is where are the segmented images located in our computer. So in my computer, I've downloaded the images from my Zenodo repository, and I store them on my desktop in this folder, segmented Rhizobox images. Voila, you have all the 35 images here that are also in my Zenodo repository, and I'm just copying the path here, and I will paste it in this text box. So these are not Mini-rhizotron images, so I just leave it on the side. I just untick this box. I keep this box checked because these are indeed images that were converted in a format that is suitable to use with RhizoVision Explorer. You can also use images that are directly exported by Root Painter. It's also an option. Then you just have to untick this box. But in this example, the images have been already converted into a format that is suitable for RhizoVision. So I will keep this box checked. You have some options, for example, to do a median filtering to try to remove some noise in your image, because when you do, especially when the images have low resolution, if you zoom in into the segmented images, you see that there might be quite a bit of noise. That means maybe some very small amount of pixels have been detected as being roots. In fact, they are background, because the model was not maybe 100% perfect or effective. So you can do a noise, you can check this box if you want to do a median filtering, and you can choose the kernel size, which is the size of the grid that will go through your image for the median filtering. I need also to provide the resolution of the images. So the images have a resolution of 240 dots per inch in this example. I want the root distribution indices to be expressed in centimeters. So I'm just choosing this unit. And I want indeed the Shiny app to calculate root distribution indices. So I'm checking this box. And finally, you can just play around here with the slider to specify, okay, what D value I want to calculate. And here is the what we call the D90 value. So it's the depth value above which 90% of the root pixel will be located. And the deep root fraction, as I said, the default value is 30, but you can change it if you want. It's the it will calculate the proportion of root pixels that are located in the lower 30% of the image. And when you have everything set up, you can just process the images. It will go through the 35 images. It's a bit slow. It's probably too slow here because I'm connected to, I have too many things running on my computer right now. Wait, I will just stop this and start again, then it's fine. I will just only keep 5 images, not 35. Voila. And I will do it again. Voila, provide the path. Should be fairly quick when you don't have too many things running on your computer. These are segmentation for RhizoVision. Yeah. Maybe I will not do the median filtering because it also takes time. 240 centimeters. Yes, you want to calculate root distribution. Voila. And when everything will be processed, you will end up with a table like this, which provides for each image the file name, the height and width of the image in centimeters, and the value for the different parameters of interest. So you have the mean routing depth expressed in centimeters, the maximum routing depth, the D90, the deep root fraction, the root surface area in square centimeters, and the relative root surface area for the last column. And once you have this information, you just, if you want to do some data analysis on this, you just download the data as a CSV file. You save it somewhere on your computer. So we'll just go save it on the desktop here. I will save. Voila. Once it's saved, I'll just exit the Shiny app. Voila. And you can then load it into your environment. You load data from text. In this case, we select the CSV file that we just created. We give it a name, data. In this case, we say, yes, the file contains headings, and we import it. And voila, you have now the images for each image, all the root distribution indices that have been extracted, and you can now start doing some data analysis on this. Of course, if you don't like Shiny apps, you can also do all these things from the command line, from the R console, sorry, because there is a function that does exactly the same. It's called process segmented root images, and you can just call this function from your script, provide the path where the images are stored, and all these arguments are exactly the same as the one from the Shiny app. You can run it and it will do exactly the same thing as the Shiny app. Voilà. So this is one way to extract information from your
Hi Benjamin, I have a question. So
yes.
you were related like the memory of the computer, or like, so if there are so since there is like um, ARP, like the command line version, do you think this package can also be used like on high computational resources in case you have very large or numerous images? Or what kind of resource do you usually use for your?
Usually I use just my normal laptop. My laptop here is quite old. It's close to 88 years old, and it needs to be updated for many features. But with with a normal laptop, there is no problem with using this Shiny app. You can analyze hundreds or thousands of segmented images in a quite short time, even if you do the median filtering to remove some noise. It's going to take a bit more time, but not that much. Here it was running slowly because I was running also this video conference setup, but it's much faster if I don't have this running.
So then you don't need like a lot of resources in terms
no,
of...
there is. I have the chance to have at the Leuphana University in Bourg, we have access to a computer cluster with a good GPU, which I use when I with Root Painter. That means when I segment the image with Root Painter, this is actually the time-consuming step, and this is I would say, the computer-intensive step, because there is lots of image manipulation happening in the background, so you need quite a good GPU. And this step, I'm doing it on a computer cluster. Once I have the segmented images, I don't need to use a computer cluster anymore. My laptop is more than enough.
Okay, thank you so much.
Okay, you're welcome. So this was the first part. So how we use Rhizoboxes to look at root distribution, and all the tools that I've shown you, the Shiny app, the R package, are freely available to you. If you have any problems running these functions or whatever, you feel free to get in touch with me, and I will do my best to help you. So in the second part now, I would like to show you how we use Mini-rhizotrons to analyze root distribution in the field this time. So we were interested in try to replicate the result from a Rhizobox experiment in a more realistic setting, which is why also we set up a field experiment. So the first question, what are Mini-rhizotron systems? So Mini-rhizotrons actually consist of two main components. A closed transparent tubes that we insert in the soil. I don't know if you can see, I have one here just next to me. So this is a transparent tube like this. Here in this example, it's 1 meter in length. And one extremity, this extremity will be inserted in the soil. You see it's properly sealed on this hand. You insert it into the soil, and this side will actually stick out of the soil and has an opening. Voila, that you can open. It's a plastic cap that you can open then to insert a camera or a scanner to take images of roots growing along that tube once it's inserted into the soil. So this type of device is usually used to non-destructively monitor root growth and also root dynamics, like root turnover, root distribution. That's why we use it mainly in our experiments at the moment, but also biotic interactions, because you can see, depending on the type of image acquisition system you're using, you can see plant association, for example, with mycorrhizal fungi. And so this is something that is also of interest for the root research community. And like every experimental system, it has pros and cons. So it's also quite flexible, let's say, in design, in the sense that you can use Mini-rhizotrons that are Mini-rhizotron tubes that are either short, for example, 50 centimeters in length, or longer. I'm aware of some experiments where they, I think it's in the Jülich Forschungszentrum, I don't exactly remember the size of the Rhizoboxes, of their Mini-rhizotron, sorry, I think it's quite long, around 5 meters, and they installed it horizontally. In our experiment, we use tubes that are 1 meter long, so much shorter. You can adapt the construction material. In our case, we use acrylic plastic tubes, and the imaging system may also change from one experiment to the other. There are usually two main image acquisition systems I will show you in the next slides. It's also quite easy to replicate in the sense that you just need these root observation tubes, these mini-rhizotrons, but they can be quite a bit pricey. So depending on the size, depending on the length, depending on the number of tubes that you buy. So it's also quite, definitely the cost is also something to take into account. And when you install these tubes into the soil, you can install it in three main ways. You can install it vertically, like this, for example, but it's not necessarily best if you want to look at root distribution, because the roots may try to preferentially grow along at the interface between the soil and the wall of the mini-rhizotron tubes. You have this kind of funneling effect happening. You can install the mini-rhizotrons horizontally, but it's really quite some work because you have to do some trenching in your site before installing the mini-rhizotrons horizontally, or you need to use a mechanical shovel to extract the soil, then put the mini-rhizotrons in place, and then fill it back with soil again. So it's very disturbed. There is a lot of disturbance if you do that. And in our experiment, we preferred installing the mini-rhizotrons at a 45-degree angle in the soil because we thought it would be the most straightforward and effective way to assess root distribution in our plots by just taking images at different depth along this tube. When you look at the disadvantages, of this type of system, as I said, they can be quite expensive. The tubes can be expensive, but it's mainly due also to the image acquisition system. Depending on whether you use a scanner or a digital camera, these are quite sophisticated equipment, so they can be quite pricey. If you use a digital camera, the viewing area is usually quite small, a few square centimeter in size. So that means that if you want to have a good picture of what's going on around your mini-rhizotrons, you would have to take quite a lot of pictures. And of course, like most experiments in root research, I mean, data acquisition is often quite time-consuming, especially if you want to have a high spatial and temporal resolution. That means if you want to take images very often in the field at at a lot of different depth, of course, this would be much more time-consuming. And like for the Rhizoboxes, it's quite difficult. It's even more difficult, I would say, to track roots at the individual or species level. Especially in our case, we work in grasslands, and we have quite a lot of different species growing in the plot, so it can be very tricky sometimes to distinguish if we have different species or not, simply looking at the morphological traits of the roots that we see in our image. So how do we install the mini-rhizotrons in the field? So this is actually a critical step, because having a very good contact between the roots and the soil without causing too much disturbance is actually very important for this type of technique to work properly in the field. So how did we proceed in our experiment? So we knew that we wanted to start the experiment in April last year, in April 2021. This is the time at which we wanted to add the seeds in our plots. And so 6 months before starting the experiment, we installed the mini-rhizotron tubes into the plot. So that, because as I said, this is quite a disturbance when you install these tubes. And so we wanted to give the soil and our system time to recover from this perturbation. And then 1 month after installing the mini-rhizotron tubes, we also installed some geotextile on top of the plots to avoid having weeds and non-target plants that grow and start producing roots around the tubes. And then after 6 months, we started the experiment. But I would like to show you a video that illustrates the different steps of this of this installation work. So In the first step, we use, as you can see, a kind of a support, like this wood support, and we use it to take a soil core at a right angle, which is a 45-degree angle. We start by taking a narrow core, just to make sure that there are no obstacles in the way, like large stones or something like this. The reason for this is that we are working in a very sandy soil, and sometimes we have really big stones along the way. And so to make sure that the path is free, kind of, we take a narrow soil core like this one. Here it went very fine. So now we can proceed to the next step, which is repositioning the support to take another soil core at the 45-degree angle. We make sure that it's properly level. And now we're using a larger soil core, which has a diameter slightly smaller than the Mini-rhizotron tube. And we insert it into the soil to create a bigger hole. Voilà. This is actually the step that is quite physical, especially if you have to use a hammer to put it in. Then you remove it. We remove the soil core from the soil. Voilà. And to make sure then that again there are no obstacles in the way, we use an old Mini-rhizotron tube like this one. You see it's full of scratches or whatever. It's not going to be used for root observation, but we insert a first old tube into the ground to make sure that there are no big or small stones that can really scratch, the plastic too much. And once we see that the hole is big enough and that there is nothing in the way, we can then insert new Mini-rhizotron tube that will stay in the plot and the experiment for quite a long time. And we are trying to be careful when we insert it. And we make sure that it's well positioned. That means we make sure that it's at the right distance between the top of the observation tube and the soil, to make sure that that when we take images at a specific location within the tube, it's always the same and consistent between the plots. So these are the main steps in a very, summarized in just 2 minutes. So sorry, it was a bit fast. And so this is how the experiment looked like once we were done installing all these Mini-rhizotrons in all the plots. So you can see here that in each plot we had 2 Mini-rhizotron tubes. The size of the our plots are quite small. They are only 3 by 3 meters, so 9 square meters. And and each treatment was replicated 5 times. That means that in this situation we have each treatment is represented by 10 Mini-rhizotron tubes. So the number of replicates that you have to install in the field is actually a trade-off between what we can afford, because buying these tubes is actually quite expensive, but it's also time-consuming to take pictures within these tubes. It also depends on the size of the plots, I think, because if your plots are larger, you might need also a larger number of tubes to try to get a representative picture of the root distribution within your plot. So there are a number of factors to really consider when you choose the number of replicates to install within each plot. And you see that we randomly allocated the orientation of the Mini-rhizotron tubes within each plot. So in some plots, the Mini-rhizotrons have been installed in a west-east direction, like here, but in other plots it was installed in the other way, from the east-west direction. And we also use, but this I will show in the next slide, but some isolating material to make sure that there is not much interference from the environment. And right after installing these Mini-rhizotrons, we also covered the area with this black geotextile to make sure that there is no, root plants growing within this 6-month interval before starting the experiment, just to make sure that there are no new roots growing along the tubes, the Mini-rhizotron tubes, before the start of the experiment. And after 6 months, of course, we removed this geotextile from the site. So, as I mentioned, we protect the tubes and the external part of the tube, the part of the tube that comes out of the soil. We protect it from light and weather conditions by using an isolating material, which is black, so it stops the light from going inside the Mini-rhizotron tube. And this is how it looks like 6 months after installing the Mini-rhizotrons. We started then sowing the plots, adding the seeds, and from now on we just let nature do its job. So now that the experiment starts, we need to take root images of roots growing along these Mini-rhizotron tubes. And there are different image acquisition systems you can use. And they're actually in the scientific literature, there are two main systems. You can either use a digital camera, which is, in this case, it's a camera that was developed by Vienna Scientific Instruments. And this is a system we use in our Mini-rhizotrons. So we use this system because the camera has a quite high resolution. That means the images have a resolution up to 2,500 dots per inch. You can really see fine roots very well, which is important for us because we have quite a lot of species with very fine roots in our plots. The drawback with this type of system is that you only have a very narrow field of view, yes, because you only see an area which is smaller than 4 square centimeters in size. That means that if you want to have a good picture of what's going on along your Mini-rhizotron tubes, you might have to take quite a lot of different images along your tube, maybe in different directions as well. So this is one system, and I said this is the one we're using, but there are other systems that have been also used in other studies, which is, it's not a digital camera, it's a scanner this time that you also insert in a tube, and it can take a 360-degree scan of your tube. That means you have a much larger than field of view with the scanner. The maximum resolution, I think, is around 1,200 dots per inch, so it's smaller than the camera, and the size of the images that you end up with are also much bigger because you see, as I said, a much larger field of view. So if you're interested in these two systems, you just feel free to have a look at their website. These are I would say the most common systems used to acquire Mini-rhizotron images, but you also have to know that a number of studies used different approaches, and I have often these two studies in mind. The study from Richard Nair was actually recently published in Journal of Experimental Botany, and the study from Camille Defresne and Colin Iverson. They both use automated Mini-rhizotrons. That means the image acquisition process is automated, and so they take much more images because it's not manual. And I think, at least for the study of Camille Defresne, the images are also at a very small spatial scale because the camera zoom really into the rhizosphere to look at root fungal dynamics in a peatland setup. So there are a diversity of systems being used. But which system should you use then if you set up a Mini-rhizotron experiment? Well, these are the factors that we considered when we had to choose what was best for us for our experiment. So, of course, we looked at the research question. What are we going to do with these images that we take in the field? We wanted to use them to look at root productivity and root distribution in the soil. So this was the main reason why we chose the digital camera. What are the size of the roots that you have to image? There are some species with larger roots than others, so it also affects you. If you have to work with very tiny roots, you might need a system with a quite high resolution, for example. Do you need to work with large or small images? How good the images need to be? What resolution you need to use? How many images do you have to take in the field? This will also affect the image acquisition time and the time you have to spend in the field to collect these images. It's not a small job. It's really a lot of work. It also depends on the, for example, if you use the scanner system, the image acquisition time is very much dependent on the resolution that you use to take your images, because you can adjust the resolution. If you want high resolution images, where the scanning time will be, of course, longer. And this is also one of the reasons why we choose to go with the digital camera, because the image acquisition time is actually quite short. In 1 or 2 seconds, you have an image. Well, of course, it focuses on a small area of your mini-rhizotrons, but it's very high resolution, and we just take more images along our mini-rhizotron tube just to get a good picture of what's going on. How robust the system is, how much autonomy you have when you're in the field, because in some situations you don't necessarily have energy supply. So if you want to take all your images in just, I don't know, 1 day, well, you need to consider this. In our case, for example, we know that the battery of the computer is just enough to take images for 1 hour experiment. And of course, how much budget you have and how much money you can put into this image acquisition system. So how do we do this, practically speaking, in the field? How do we collect root images in our experiment? So we have this following protocol. So this is the mini-rhizotron tube installed at a 45-degree angle. And along this tube, we insert a camera, the camera that I showed you a few slides ago, and the camera is fixed to an indexed handle that we can just use to position the camera at very specific locations within the tube. And usually we work in pair. We have one person that who takes the images with the computer, and we have a second person who moves the camera along the tube. We take along each mini-rhizotrons 18 images. 1 image roughly every 2 to 3 centimeters. So we don't have overlapping images. That's important. The images are independent from each other. And we do this for every single mini-rhizotron in our experiment. So in this case, in our experiment, we have 40 mini-rhizotrons. We take 18 images around each. So we end up at the end of the day with 720 images per session, which represent between 3 and 4 hours of work, depending on how fast we are. With the digital camera, I have no idea how long it would take with the scanner. So this is the type of data you end up with. So this is just a time series of root images taken at different depths within a tube. And what I find really interesting with this type of images is you can see new roots appearing, but you can also see over time roots disappearing. You see, for example, this root here is still quite alive in October 2021, but in February 2022, a few months after, you can see that the roots start to disappear. You can also see the diameter of some roots really shrinking over time. So you can get from this quite a lot of useful information. For example, if you're interested in root turnover or something like this. In terms of how often do we take pictures in the field, this is also something that is really important to consider when you do this type of experiments. So we decided to take images every 2 weeks, so twice a month during the growing season, which is typically between April to September in our region, and between October and March, so during the autumn winter, we only take pictures once a month. So that means per year we collect around close to 13,000 images that we need then to be analyzed. So, but the analysis of these images has comes with a few challenges. And if you look at these challenges here, you have, I just illustrate a few challenging situations. So you have here a situation like this, where you have lots of roots growing really next to each other, which is not easy. For example, if you want to detect roots, individual roots, to try to get a good estimate of the root length density in your image, well, this situation is very tricky, because if you have to train a neural network with Root Painter, it's likely that some of the roots will be, for example, this feature here. It's not just one root; it's actually 3-4 roots growing next to each other. But it's likely that the network will detect them as one big root. And so, of course, you would underestimate the root length density in your image if you do that. But I think you can still get a good approximation of the root surface area, for example. So this adjacent root problem is challenging. You have also images where you have lots of root hairs and fungal hyphae, for example, which you do not a priori want to detect, which you just want to focus on the roots, which is okay. So you just want to segment the roots in this image and not the root hairs, for example. In some tubes, you have some scratches. This is unavoidable when you install tubes in the field, especially in sandy soil. There are a few scratches, and we need to train a root painter model that is able to segment the roots without segmenting these scratches. And you also have situations like this where you have some drops of water, especially after rainy days, on the outside of the tube. And what you see here is actually not a root, it's just a reflection of a water drop. So this type of feature, you just want to also train the model to avoid detecting this as a root. So these are complex and tricky images to work with, but we use a very similar image analysis pipeline as the one that I showed you for the Rhizoboxes. That means we start with an original image like this one, which is not too problematic. It's kind of an easy image to analyze and segment. We train a neural network model with Root Painter to be able to segment this image. And once we have the segmentation, the segmented image, we try to extract information from it. And to do this, we mainly use RhizoVision Explorer, which is an open access and open tool that was developed by the team of Larry York in the USA, which allows you to estimate, for example, the total root length in an image, the number of branching points, the branching intensity, or the diameter of the roots, etc. But you have to know that for our when we trained a neural network with Root Painter, we of course we were aiming to train a model that is able to detect the center line of the root, not necessarily the totality of the root, but really its center line. Why? Because when we have this type of images, we really wanted to try to detect each root independently. And so we wanted to focus on the center line of the roots and not the totality of the roots. And so in our case, the diameter provided by Root Painter, by RhizoVision, is probably not very useful because we very likely underestimate the real diameter, which is also a common thing when you use Minirhizotron images anyway. And this type of pipeline it's not only used by us, it's also used by colleagues in the Jülich-Forschungszentrum, but also other colleagues in Germany. So it's a very well known, I think, established pipeline, image segmentation of Root Painter and feature extraction using RhizoVision. But I would like to tell you a bit more about the strategy that we developed for image segmentation and to train a Root Painter model to process all our images. So we had, after 25 time points, we had close to 18,000 images in our computer. And we thought, okay, now it's time to see what's the story behind this data. And so we started, okay, how do we then train a neural network with Root Painter on these images to get good segmentations? And we worked mainly as a team of two. So we were two people that were involved in this task. And so what we did is that we created two different sets of training images. One that will be used by scientist number one and one that will be used by scientist number two. And each training set consisted of the images taken at two different time points. And the time points that were selected were different for the different users. So we had two different training sets. And each training set consisted of two time points, so a total of close to 1,500 images. Then we used Root Painter to create a training dataset with these different sets of images. And we did this by asking Root Painter to randomly select 2 subregions within each image in our training set. So as a result, we multiply by 2 the number of images, and each user started the model training and the annotations with 2,880 training images, which were smaller in size. They were around 800 pixels in size. 800 pixels by 800 pixels. Then we used Root Painter to annotate these images and train a model in the background to be able to correctly detect the roots in our training images. And we did this for quite some images. We decided to screen at least 500 images from our training dataset. And after 500 images, we noticed that we didn't have much of an improvement in terms of model accuracy, so we decided to stop. And as a result, each user, each scientist ended up with a Root Painter model. The blue user with model number 1 and the yellow user with model number 2. So we had 2 different models, and that could be compared to each other to see if there are some differences with image segmentation. But we wanted to push this comparison a bit further, and we wanted to see, okay, what happens if you pool the annotations from both users together and you retrain a new model using this extended annotation dataset? Because there is this kind of rule in machine learning that the more data you provide, the more annotations you provide, the better your model will get. And so our hypothesis kind of is that if you combine annotations from different users and retrain a model, you would end up with a better model than the model obtained by each user independently. So this is model number 3 that results from combining annotations from different users. And you also use another approach, which is called model averaging, where when we segmented our root images with Root Painter, we didn't select only one model, we selected 2 models. And so the result, the outcome, is an average of the 2 models. So we had 4 different models to compare. But because we didn't have any ground truth data for this analysis, we were just limited to comparing the results of the model to each other and see how they compare to each other. But this is a graph showing for model 1, so trained by one of the users. It shows the evolution of the accuracy of the model as a function of time, or as a function of the number of annotated images. And you can see that over time, when the number of annotated images increase, the accuracy of the model really there is an upward trend in this curve that is quite obvious. And the accuracy here is just a number between 0 and 1 that gives you the proportion of pixels in your image that have been predicted correctly by your model. So if you are at 100%, that means that 100% of the pixels in your image have been predicted correctly by your model, which is, of course, what you want to tend to, I would say. But after 600 annotated images, we didn't notice any more improvement for the model. So we decided to stop there, and we decided then to move to the next phase, which is comparing different model outputs. Then what we did with these 4 different models, we used these 4 models to segment a new set of images that were not used for the training, either by user 1 or user 2. We wanted really to use independent sets of images. And we then compared the results, the root length density that was calculated using the segmentations from the different models. And you can see, for example, if you compare the models obtained by the 2 different scientists, by the 2 different experimenters, you can see that the results are highly correlated to each other, which is certainly what we would expect, but they don't exactly agree with each other. That means that the model 2 tend to underestimate the total root length in an image, especially when you have lots of roots in the image. But of course, when we look at I was quite surprised because I was expecting stronger differences between the 4 models. And so, as all the models as all the results from different models were quite strongly correlated to each other, we decided to inspect the images that seem to be the most problematic. That means the images where we had the most differences between models. So this is an image, for example, that is not problematic at all. You see that it's quite easy to segment. The roots are appear clearly, and you can see that the 4 models, root painter models, actually perform quite well. All of them. Some detect a bit more background than others. For example, these water drops have been a little bit detected by model 1 and 2, but not much by model 3. So you have a few differences, but not minor, not major differences here. But in this situation, you can see that you have big differences in model performance because you're in model number 2 in this. When you have this complicated image, clearly underestimate both the total root length in the image, but also the surface area, because this part here of the root was not correctly detected. Model 1 seems to do a slightly better job, but it's still not perfect because you have also some of these either hairs or fungal hyphae, I don't know, who seem to be detected. But what's interesting is that when you combine the annotations from the different users, from user 1 and user 2, and you end up with a new model, you see that the new model that you get after combining this annotation is actually much better. It's actually much better. That means we have many of the roots detected. We have only very few background being detected. And this the 4th model that results from this model averaging strategy didn't really perform as expected, because you see this segmentation in the center here is really not good. It's much better when you combine annotations from different models. And you can see again this result from this also quite complicated image. Again, model number 2 seems to slightly underestimate the amount of roots in the image, and model number 3 and 4 in this situation had comparable performance, although here model 4 you see kind of do not detect well the root on the upper right corner, but the model number 3, which is based on combined annotations, is actually much better. So based on this manual inspections of a number of images, the most problematic ones, we decided to go for model number 3 because it's this model consistently provided the best segmentation in most of our images. So now that we have a model, neural network model that we can use in Root Painter to segment all our images, we did the segmentation and we ended up with thousands of segmented images from which we want to extract information about that could be used afterwards to look at root distribution. And to do this, we use RhizoVision Explorer, which, as I said, is a open access and free software that you can download from this website. And I strongly recommend you to have a look at the Phenom Force workshop from Larry York from last year, I think. Larry really described very well how the software works and so on. But I would like now to take the opportunity of this workshop to show you how we are using RhizoVision to analyze or segmented mini-rhizotron images. And you can also download a dataset consisting of, I think, of 100 segmented mini-rhizotron images. You just go to my Zenodo repository, you just click on this link, and you download the segmented mini-rhizotron images here. And you will then be able to try RhizoVision. Of course, you will need to install RhizoVision first, but everything is very easy to install it, and you can download it directly from the website. Okay, so I will show you then how we use RhizoVision to analyze our mini-rhizotron images. So first I will open RhizoVision, voila, like this. So it's a very elegant graphical user interface, very simple to use. I will guide you through the different steps. So the first thing we're going to do is we're going to load an image. So we go to File, Open an image. And voila, slow. I'm going to my desktop and I go to the segmented images, Minirhizotron images. Voila, that I do. These are the images provided on my Zenodo repository. And I select, for example, an image like this one, quite complicated. So this is the segmentation output that we get from Root Painter. So the first thing I'm going to do is to adjust the parameters for the thresholding of the image. That means we are going to select only the root pixels in the image. So we have to use the broken root mode, obviously, because the roots are not connected to each other. And I will provide the resolution of our images. So we have images that are 148 pixels per millimeter. This is the value that I got using image chain. And here, this is the image thresholding level. So usually RhizoVision is a software that was developed to process gray level images, where each pixel in your image will have a value comprised between 0 if the pixel is pure black to 255 if the pixel is pure white. And you can see here, we have the output of Root Painter is actually more of a binary image. It's not a gray level image, it's a binary image where a pixel can either be black or white. So the thresholding value that you choose here doesn't really matter. Of course, it cannot be 0 because then you will just detect nothing, but it can be any value between 1 and 255, basically. Which is, of course, not true if you use gray level images. But for these binary images, I will just set the thresholding value to 200. It will work. So then I will use this, often use this option to filter non-root objects in the image, because you see that you have a little bit of noise in the image. Maybe those are probably small root hairs that have been detected, or maybe some small particles. And so you just have to specify, okay, what is the maximum size of the objects you want to filter. And here I'm just specifying that I want to filter all the objects that have a size smaller than 0.05 square millimeters. It's very small, but don't forget we are really zoomed in here into very fine roots. So you can preview the segmented image by clicking on this button. So you see here that most of the small objects disappeared, and I'm mainly left with roots. It's important to really play around this value, because if I use something that is parameter is too big, for example, 5 square millimeters, you see that we are start removing roots from the image, which is definitely not something we want to do. So I figured that the 0.05 value was optimal for our Mini-rhizotron images. I do not recommend to use the fill holes in root objects, because you see this, for example, this type of hole will be filled with black. I can show you how it will look like. Voila, you see, if you fill the holes, then you get this weird pattern. So I would not use this option with RhizoVision. And you can use the enable edge smoothing option. And usually I use it to try to smooth the edges of the roots. Once you're happy with your setting, and once you have a segmented image that looks okay to you, you can go to the feature extraction menu. And this is where you set the different root diameter ranges for which you want to extract information like length and volume and so on. But in our case, I mentioned, we mainly focused on training a neural network to detect the center line of the root. So the diameter information for us, in the case of our experiment, doesn't really matter. And so we remove the different root diameter categories. But if you're interested in extracting root diameter information from your images, then you can specify, I think, up to 10 diameter classes. So let's have a look at what RhizoVision does with this setting. So we ask now RhizoVision to skeletonize the roots in our image. So you see that the skeletonization went well. All the roots have been detected. And these are the features that RhizoVision extracted for this particular image. And you see, for example, that the total root length in this image is around 370 millimeters. But I would strongly recommend you to check your images to zoom in to see if the segmentation, if the skeletonization, went well. And you can see here there are a few things that are not so great, in the sense that you have these fake lateral roots being detected. That means you have small bumps kind of, and the software interprets this as possibly a lateral root. In fact, there is no lateral root here. So this is problematic. And you can use, to get rid of this fake lateral roots, you can use this enable root pruning option. If you look at the help guide for RhizoVision Explorer, you can see that the root pruning is recommended to delete false root segments where the tips are longer than the diameter of the parent root, plus the additional pixel value that you supply in this root pruning threshold box. So here, for example, I will use a value, I think, of 10, just to see how it will look like. You reanalyze the image. Boom. And now you see that these fake lateral roots are not there anymore. So this root pruning option is actually very relevant because it can make quite a big difference. You see that now the total root length in the image dropped to 346 millimeters and not 370 anymore. So we were overestimating the total root length in the image before. And when I look at this image, I don't see big problems anymore. I would say it's a pretty nice skeletonization. And I would repeat this test for a couple of images, let's say, for example, 10 different images, to make sure that the method that I just created in RhizoVision works well for a range of segmented images. And when you are happy with your method, you can then use the batch processing option in RhizoVision to analyze all the images in your repository in just one step. So I will do it here. So you first have to provide the information on where the images are located. So in this case, it's in this folder in my desktop, segmented Mini-rhizotron images. I select the folder. We have to decide where we're going to store the RhizoVision outputs. So I will just create a folder here for the results. I select the folder, and RhizoVision, after this batch processing, will create two different CSV files. One called features. It will be a CSV file that will store all these parameters calculated by RhizoVision for each image. And you will also have a metadata file, which is extremely useful because it will save all the information about the method that you use in RhizoVision. You have also the option to save either the segmented images or the processed ones. So the segmented images are just, the black and white image after segmentation. And the processed image, it's this image that you have over here. It's the segmented image on top of which you overlay the skeletonization. And so usually I save the processed image just to, if later in the analysis we find a problem, we can just go back, open the image and see, okay, there is no root in that, for example, in the image, or I don't know, maybe the skeletonization didn't went so well, and that's why we have this value or something like this. So I always like to save the processed image. Then once you're ready, we can just start the analysis. So here we have 100 images in our folder. This is quite, RhizoVision works quite well. That means you can literally analyze thousands of Mini-rhizotron images per hour. Here again, it's quite slow because my computer is running other things in the background. But this is how you run the batch processing mode. And when all images will be processed, 100%, it will stop. And then you can just quit the batch analysis. So I will just do this now. I will not wait that it comes to an end. So I quit, voila. And here I have, for example, an example of feature file that is provided by RhizoVision, as well as the metadata. So open, for example, the metadata well, you have all the parameters used for the method, which is very useful when you have to write the method section of your paper, for example. And all the features are stored here, but I will show you how we open this in R a little bit later. So, so from all the parameters that RhizoVision extracts, what are the ones that we mainly use for our experiment? So we mainly use actually two main parameters, the total root length in the image and the network area, which is the kind of root surface area in your image. And these two parameters in all images are actually really strongly correlated to each other. So in the next graph, I will just show you the result for the based on the total root length, but I could do exactly the same analysis using the root surface area. Just keep this in mind. And usually I would say they provide information that is quite complementary. Total root length can allow you to calculate the root length density, which is related to soil exploration by roots. But the network area allows you to calculate the root surface area in your image, which is, according to a recent paper published by Richard Nair in Journal of Experimental Botany, is a bit more, is a better correlated with biomass, for example, with biomass than root length density. So when we do data analysis in our research team, well, we have this project data. That means we have all these images from which we extract a lot of different parameters. And comes a time where the different team members want to use this data for their own project to answer this question. And so these different team members, they write some pieces of R or Python code to analyze this data. And the question that I had at the beginning of this project is, how do we make these different pieces of R or Python code kind of compatible with each other? Because different users, when you work, for example, with students, when you work with PhD students and postdoc, well, we all write code in our own way, and sometimes we have our way of naming variables, etc. So that means that the code that is written by one person is not necessarily directly compatible with the code written by another one. And in this case of our project, I wanted to make sure that we had as much compatibility as possible between different team members to make, the collaborative work as efficient as possible. And if you want to do this, but standardization is really key. If you want to make the different pieces of code written by different people compatible. And so to do this, in this R package that I developed for this project, this Poem Demo R package, I also coded a very simple function to load RhizoVision data in R. And it's a very simple function that has only two arguments. You provide the path to the CSV file containing the features extracted by RhizoVision, and there is an extra argument to know, okay, are these data related to your Poem project or not? And in our case, Poem is a very, personal project, a research project in Leuphana. If it's not, the data are not associated with this project, you set Poem to false, for example, you load the data, and if you look at the data, you have the first column is just the file name, which is useful. But in our case, this file name is actually very information rich because it contains the name of the experiment, the tube number in which the image was taken, the location along the tube, the date at which the image was taken, etc. And so the idea of having this function here, when you set Poem to true, in our case, it does exactly the same thing. It loads the data from RhizoVision into R, but this time it adds a number of variables to the data file, which are of high relevance to us. So we have the name of the experiment, the plot number where the tube was located, the treatment associated with this plot, the replicate number, the tube ID number, the location along the tube, the depth at which the image was taken, expressed in centimeters, the date and the number of days since the start of the experiment. So this is also information that is very useful when you plot the results. The name of the person, from the abbreviation of the name of the person who took the images, and finally you have all the features that are extracted by RhizoVision. But the benefit of having this function and this R package that is used consistently by all team members is that we have then a very consistent way of naming the different variables, and we all start with the same data set. So that means that we can just very easily exchange pieces of code. We don't have to start spending time renaming the different variables, and we just exchange code, run it, and it just works. So it doesn't look like something big, but it's actually quite a time saver. Okay, so this was done. So, and at the end of the analysis, once you have your data table in R, this is the type of information we extracted. So this is a graph showing the evolution of the root length density in the images over time since the start of the experiment. And you can see actually the two growing seasons quite well, the first growing season at the beginning and the second growing season in 2022 here. And these are model fits that were created using a statistical framework called generalized additive models. We used GAM models in this situation because we have an obvious nonlinear pattern in the data. And if you want to take this into account, GAM models are actually very interesting to use. But how do we use this root length density estimated in each image to assess root distribution at the community level? Well, the approach we're using in our experiment is that we calculate for each cube, we can calculate the mean routing depth. How do we do this? But remember, I told you that along each cube, we take images at 18 different locations. So 18 different depths. And for each of these depths, we have a root length density value that is given by RhizoVision Explorer. And so what we just have to do is to apply this formula where each depth value is weighted by the root length density in the corresponding image. And then you divide the numerator by the sum of all the root length densities measured across depth. And this will give you an estimate of the mean routing depth. That means the depth value above which, on average, 50% of the roots are located. And when you do this, we can produce this type of graph. So this is the different treatments that we have in our experiments. Not that much important here. We just have situations where all plants are sown together or where one functional group is sown before the others. But we have this graph where you have the relationship between soil depth on the vertical axis and time since the start of the experiment. And so you can see, and the raster image that you have on the plot is actually model predictions given by your GAM modeling. And you can see that you have the curves here that show the evolution of the mean routing depth over time at the plant community level. And you can see that in two situations, for example, when forbs or legumes are sown first, we have actually two root hotspots toward the end of the experiment. We have high root length density at the bottom, but also at the top, but also at the bottom of the soil profile, which explain why the mean routing depth of plant communities start to drop towards the end. But this is exactly the type of graph we were looking for when we started the experiment, because we really wanted to see how the order of arrival of plants really affect the vertical root distribution at the community level. And this result are quite fascinating to us, but it's still quite preliminary. But this is the type of output you can get using this Mini-rhizotron images to look at root distribution and how it changes with depth and over time, which I think is very interesting. So to just end this presentation workshop, I would just to mention a few limitations of the approach that we used. to analyze our images. So the first thing is that it's very hard, it's still quite hard to track the birth and depth of individual roots, which is actually something important if you want to estimate root lifespan or root turnover. And in this situation, if you're really interested in this, some manual tools like Rootfly or Rhizotrack may be needed if you want to extract information at the root level. But this is definitely something that will require additional development. But this is certainly of interest when we look at Minirhizotron images.
Benjamin, do you mind if I interrupt you a little bit?
No,
because I think there's been a bit of discussion behind the scenes about all the interesting information. It's quite a sequence. As a soil scientist myself, with a tremendous interest in roots, we have been discussing about the use of this for different crop plants and things. But from my side, it's more like, suppose you have tried different types of soil. So it's I can see some limitations in the actual logistics of deploying the Mini-rhizotrons in the soil. Have you encountered particular problems with different types of soils, or?
Not in our case, because I think we are pretty lucky in Lüneburg in a way. It's a nice place to work with roots because the soil is very sandy. So we have a very sandy soil, so it's quite easy to install these Mini-rhizotrons. These Mini-rhizotron tubes. Of course, there are sometimes a few stones that make us impossible, and we have to just shift the position of a Mini-rhizotron a few centimeters. That's not really a problem. I've never worked or developed this type of system, for example, in very loamy or very clayey soil. And there are definitely some challenges with some additional challenges that come with this type of soil for sure. Especially, the development of cracks or something like that can really affect your results when you use Mini-rhizotrons. But so far, I've been lucky, mainly sandy soil.
And so this is a relatively new approach. It's really fascinating, the idea of seeing in real time. You can take sequential time of how the root is actually developing in depth. And of course, we know that the root depth and the development is important for the crops to reach resources, so on. But obviously, when you are deploying the tube, as we have seen now this nice image, the root is growing also around the tube. How much the disturbance of the tube where the camera is inserted? So what is the balance in between the disturbance you're introducing in the actual growth of the root, which obviously the advantages of filming in a way this growing?
I think it's a very good question, and it's, I think, something that is not that easy to measure, I believe. I know that there are some studies that compared, for example, different type of material when you use glass tubes or plastic tubes, and they noticed that actually the roots behave quite differently. For example, the phenology was a bit different, the coloration was a bit different. I think the diameter may also have been a bit different, but clearly the material that you use to build the tubes may already have an effect on root growth. And when the roots grow along the tube, of course, it's probably a less resistance than growing inside the soil, because I would expect the interface between the tube and the soil to be maybe slightly less resistance than just 100% soil. And see, this probably could affect, for example, root diameter, for example. But I've never tried this. It's just purely hypothesis. But yeah, it's likely, although I do not know exactly what would be the results, but it's likely that the disturbance that is caused into the soil by the tube might slightly affect the root morphology, for example, or root growth. But to be measured and tested.
Obviously, we can see immediately the advantages of a real-time observation.
We haven't measured this in our experiment.
And my last question for you, and then I will let you continue, of course. I would like also ask you, for my own sake, to actually kind of recapitulate very quickly the main, because it's been, particularly those images, so with your pipeline, these nice images that you have show at the end. It's a really nice reconstruction, and I think later on I would like to discuss that with you. Okay. But from my side, it's also a lot of people are interested in root phenotyping for the impact that it has in the plant. What about, do you think there is a possibility to gather some information on actually the impact of the root as it grows in the soil structure from these images?
On the soil structure?
Or, in the way that is a brick in the soil, and because it's very nice images.
Yeah, could you, for example, give me an example of traits or something?
Well, things like, the kind of the growing of the root is developing pores and aggregation.
Mm-hmm.
Things like that.
I don't really know how because it's like probably like the question is probably related to your previous question when you mentioned that does the root grow in the same way along the tube, where we can probably ask the question, is the soil also comparable next to the tube or a little bit more far away from the tube? So I think I would say usually we try to have a kind of mixed approach in our experiment where we have mini-rhizotron tubes to look at root distribution, and we know the limitation of this type, but we're still doing some destructive measurements using just simple soil coring in the same plots for different reasons, but also to validate the results, the root distribution results that we have with the mini-rhizotrons. Of course, we cannot have the same temporal resolution with root coring because it's just one snapshot of the root distribution. But this soil cores could also be used to look more at soil aggregation, for example, or stuff like that, I would assume. I would try to combine different techniques. I would not try to have one technique to do everything. That's my to-go message.
I totally agree with you. And just one last thing I was thinking. So what I see in the advantage of your pipeline, personally, is that you eliminate a lot of artifacts, isn't it? I think you really polish the analysis to a point where because one thing that is a lot of skeptics in the analysis of image, and I encounter that a lot when I go and present my results with RhizoVision Explorer, is the possibility of artifacts, all the bidimensional imaging and so on. And things like, for instance, in the depth of the angles, I think your pipeline seems to solve a bit of all these artifacts. What would you say?
Well, it's never perfect. By artifact, you mean the depth of the depth of field of the image, or what?
Well, I think, for instance, what all these root hairs that kind of artifacts like overcounting root hairs, or even when it comes to measure angles and depth.
Yeah, and it's obviously it's not perfect. So the advantage of mini-rhizotrons is that it's non-destructive. You can do it over time, and so you have quickly a very rich data set. But the analysis is never perfect. I mean, and I try to show in this presentation, to be honest, and show also situation where segmentation is not perfect, because there are images where it's really good, and there are some situations where you have a few root hairs being detected and so on. But it's really a trade-off between accuracy on one hand and throughput on the other. And something that I noticed when I work with, because I worked on this data set with different people, already 2 students and also researchers involved in the project, and different people trained different root painter models. And then we ended up, of course, with different models with different results, but different segmentation results, I mean, when you look at individual images. But they were still close to each other, of course, but there are slight differences. But when you look at the results, the root length density over time or the root distribution, it's really similar. It's really, really similar. So it gives me trust in this pipeline. Of course, it's not perfect. We know that. We did our very best to have a good segmentation model. And RhizoVision Explorer does an amazing work at extracting the features we want from these images. But there are always a few individual images that are problematic. And
that's what we try to sort. Thank you so much for your feedback. I will now let you continue, Benjamin. Thank you so much.
No problem, Maria. Actually, this is something that I mentioned, that we also use root coring, soil coring, in our plots to also look at biomass distribution and to try also to validate the root distribution results we have with mini-rhizotrons. Again, the parallel may be interesting. We don't have data at that moment to show, but we have certainly done that. And another frontier is really to try to be able to distinguish roots of different species. It's still very difficult, especially when you work in, like us, in species-rich grasslands with a lot of different species, a lot of different root types, and so on. There are a few techniques that have been used in the lab, like using, for example, transgenic plants that express fluorescent proteins in their roots, for example, to try to be able to distinguish under different light conditions. You can distinguish the roots based on the fluorescence, the roots of different species based on the fluorescence they emit. But of course, this is a very limited approach, and you cannot really use this in the field anyway. So there are certainly development that can be done to try to make it a bit easier to try to look at what's going on at the species level. And of course, image acquisition is still quite a time-consuming process, but I would strongly recommend you to have a look at recent papers that came up recently and that propose and developed affordable robotic mini-rhizotrons. And one that just came out recently is the one from Richard Nair in Journal of Experimental Botany, and it's a good example of what can be achieved with robotic mini-rhizotrons. I would like to also, if you are interested in this topic and if you are interested in installing mini-rhizotron techniques in the field, this is definitely a very interesting read that I would strongly recommend. This paper from Boris Riwald and Jonathan Epfrat, who guide you and provide you very interesting tips and tricks on how to install mini-rhizotrons in the field. It's kind of a classic. And of course, you saw it in my presentation, and this is something that I would like to highlight. The power of open science, because well, I'm not a computer scientist, and in our research group we are not computer scientists, but the image analysis pipeline that we're using to get information from our images heavily rely on these two tools: Root Painter for image segmentation and RhizoVision Explorer for feature extraction. And these two software tools have been developed independently, but the developers really made their work open access to everybody. It's been available to anyone. And I think it has a huge impact on the root research community. At least it has a huge impact for us, because it allowed us to obtain the result, it allowed us to test our hypothesis, I would say much more easier, but also much faster. Without these two tools, it would have been very, very difficult for us to get to this point. It would have been much more time consuming. So this is, I think, a very nice example of how open science can really improve root research. So I just wanted to highlight their contribution. Indirect, but still very important. And last but not least, huge thanks to Ines Alonso Crespo, who is here, who is the main researcher working on this field experiment where we set up the mini-rhizotrons. And she's actually the one who spent most time in the field to collect the root images, train root painter models, segment the images with RhizoVision. So most of the work that I've shown is part of our PhD. So big thank you to Ines, and also to all my other colleagues from Leuphana and the students that were involved with us in this experiment. So you are more than welcome to contact me if you have any more questions, of course, or if you have troubles running some of our analysis pipeline. And if you have any more questions, just let me know.
Thank you, Benjamin, for the great presentation. It was very great. And I think Felipe is in the background. Do you want to come, Felipe? Or? Okay.
Sure.
Hi.
Hello.
But hi. So it was a very great presentation. Thank you, because you showed how to apply really different tools. And I think the folks here, they have also a question for you, right?
Yeah. Go for it. Oh, Maria, you can go first. But before, Benjamin, such a great work, man. You did awesome. Yeah, you did an awesome combination between open source tools, and you have your biological question, and then you can really apply the phenomics in a really way that you are getting results that really can make difference. So thank you so much for showing and sharing with our community today. Good job.
Yeah. I agree with Felipe. Like, it was a great presentation. I normally work with above canopy traits, and I could learn a lot from you today. And I was taking all my notes here. Yes. And my question is, I had the chance before to work with flood tolerance, and again, it was above canopy traits. I was wondering if you have heard or if you would recommend using rhizotrons for this type of project, for being able to check the development of the root. And of course, it will depend as well as the soil. It will depend on the soil, as you mentioned before. And normally the soils that I had to work with was a kind of clay soil. So
it's a bit more difficult to install. I've never tried. I'm sure there are probably some teams around the world that use mini-rhizotrons or try to use mini-rhizotrons in clay soil. So I would definitely reach out to them. When you mentioned tolerance, what kind of tolerance are you talking about?
yeah, we were, since I'm from the plant breeding program, we were trying to select genotypes that are flood tolerant, soybean genotypes that are flood tolerant.
Yeah.
To
I think there is, because you mentioned that you work a lot on canopy phenotyping, and actually I find it very interesting to combine both root and canopy phenotyping. We often call the roots the hidden half, and the mini-rhizotrons actually are quite a good and interesting way to get a hint of what's going on below ground. Especially if I would assume that you would be interested in root distribution, if you're interested in intolerance to flooding, because maybe it can also explain why some varieties or cultivars are more tolerant than others. So I think the power would be really to combine mini-rhizotron phenotyping, root phenotyping using mini-rhizotrons with additional measurements taken above the ground at the canopy level, and have this data coming together to try to explain why some varieties or cultivars are more tolerant than others.
Yes,
I think there is a lot of potential in there.
I agree with you. And have you ever had the chance to work with different crops?
So far, I've mainly worked in a grassland ecosystems, so managed systems, but not in crops so far.
Okay, good. Thank you.
But I know other teams that work on with mini-rhizotrons in crops, and some settings are very impressive. Some field site with really thousands of mini-rhizotrons, really really impressive.
All right.
My turn. Actually, I have two questions. Did you try to put your equipment in soil in a vertical and not, or only on 45 degrees? Because in my imagination, if you are in 45 degrees this way, this upper part is not covering well the amount of roots that we have on the first part of the profile.
Yeah.
Compared to the vertical position.
Yeah, you mean for the very first centimeters of the soil, you mean?
Exactly, yeah. And from 0 to 20 centimeters, maybe, depend of the crop, yeah.
Yeah, I mean, you might be right in there. We didn't try. We installed directly the mini-rhizotrons at 45-degree angle, and we wanted to have all the mini-rhizotrons installed in the same way. So we try to avoid having vertical ones and so on. What we see from our data is that we have really a hotspot of root density in our grassland around 15, 20 centimeters. So that means above that we have not much less roots, which could be explained maybe by what you just mentioned, that maybe the roots do not develop well. Between the roots and the tube is not maybe so great.
Yeah, because for example, if you have the amount of roots this way, right, and then you capture these, so this first part here is way far from what the camera is capturing compared to this middle part. Then I don't know if it gives us an illusion that actually the great concentration of roots actually is 15 centimeters, but it would be from 0 to 10. That's just my question, because I was kind of thinking, how is going to be the results if put in the vertical position?
I would be interested in that too, actually, because I would be interested. We know that the position of the mini-rhizotrons can have an effect on the results, and we went for the more classic approach to look at root distribution, having the 45-degree angle. I don't have data to compare with vertically installed mini-rhizotrons. Yeah, if you're interested, you're more than welcome to.
Okay. Yeah, that seems a good idea.
No.
I don't know if the other guys have more questions.
I have one question that maybe is related with Maria's question. So I think like I have been always working with crops, especially with tree crops, and also in the breeding perspective that we have really to screen a lot of trees or a lot of individual at the same time. Or so my question, so one of the things of using camera is also to have a large scale phenotyping. So what is your opinion regarding that? So do you think your method can be also used to do some type of large throughput phenotyping? And I think, like, for example, so in your case, yours is more like from an ecological point of view, right? So but imagine that you have many trees or like high intensity, no, many trees, let's say high intensity field with individuals. So you have different roots from two plants that are adjacent one to another. So do you think we need to put one of the per individual, or we can also access different individual from both sides of the two? Do you know what I mean? So do you
Yeah.
think that can like translate this approach to be more large scale for high throughput phenotyping?
I think, would you be interested, for example, in this, and when you have, because I agree to, it sounds like even if you have only two species, if the roots of the two species are kind of entangled and grow along your mini-rhizotrons, with the two species you have in mind, you would be able to design to recognize visually the roots of the different species, or the roots would be very similar?
Yeah, so if I'm working with one individual, yes. But the difference when we are talking about breeding plots, I think that the individuals, they are planted in a certain distance. So
Yeah.
of course, it depends on the root system of the plant, because I guess the tree, they are very large, maybe the cereals or the annual, they are smaller. But so it will be, well, there will be also like some weeds, so it's complicated. But the thing is like, it will be nice if this system can, because root phenotyping, it's always very challenging from breeding perspective, because you need to destroy the plant almost to phenotype it, right, most of the time. And so I was wondering if it would be very interesting to use the same technology with a camera that can actually rotate somehow and take picture from two plants. And so, but I don't know if that it's possible. I don't know.
You mean camera, the thing?
So because when you put the camera, so if I can take picture from two sides, and so I can have,
Yeah, I mean, you can technically take pictures from any direction you want. You know, at the moment, you just look at the top. But
still like
I would be surprised if you really have only one individual growing on one side and the other in the other, because I think it would be always a mix of the two. I think what's important to consider is where you put the mini-rhizotrons in your plantation. Because it's like when you put mini-rhizotrons, for example, in a crop, you know that you will have different results if you put the mini-rhizotron between the row or within the row, for example. The will be completely different. And in your case, you might have a gradient. If you put, for example, you have two trees separated by 10 meters, for example, and every meter you put one mini-rhizotron, so you have 10, but you probably have a gradient where the mini-rhizotron that is closest to the species A will have more roots of species A, and the other is true for the other side. So we might have a gradient. But the nice thing now, I think, with these mini-rhizotron techniques is that we reached a point where with these tools, this root painter and RhizoVision, especially if you just want to extract information at the image level, not at the root level at the moment, but at the image level, the bottleneck is more now towards image acquisition. That means acquiring the images can be more time consuming than not getting information from them. Of course, in the context of the pipeline that I just described, for the analysis that I described. So I think now that there is this slight shift in bottleneck.
Very interesting. Yeah. Do you guys have more questions for Benjamin, or we should just thank him and move on? So,
Yeah.
well, thank you so much, Benjamin, really, for the great workshop.
Thank you very much for your invitation. It was a pleasure to be here.
we could see now a complete pipeline because we have Larry York, right? Last workshop also was part of your pipeline. So thank you so much for giving this wonderful presentation.
You're welcome. My pleasure.
Thank you. Bye.
Bye.
Wait, let's stay here for the introduction of our next speaker. I think, Maria, do you want to take it?
Yes, our next speaker will be Peter. He will be talking about phenotyping root hairs using microscope images. And I hope you guys can join us next week. Oh, not actually, it's not going to be next week. We're going to have a break of 1 week. So we're going to be sending the registration link. And I hope you can join us.
Yeah, so watch out to our Twitter and your email, and so you can register also for Peter webinar. So thank you so much for joining today, and we hope to see you all, and thank you all of you for working with us. Bye-bye.
Bye. Bye.